Ascomycetes reproduce asexually by forming conidia. Basidiomycetes also reproduces asexually only, but what is the difference here? It is either by budding or by formation of spores. Write briefly about the steps involved in sexual cycle of kingdom fungi. There are 3 steps. One is plasmogamy, karyogamy, meiosis. Most of the millennials do not have a cell wall, they lack a cell wall, but rarely few of them they have a cell wall, but it is non-cellulosic. Hello everyone, a warm welcome to this session on revision class for first PUC Biology. I am Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, it is a continuity of the previous session, wherein in the previous session we learnt about some of the important topics, that is the synopsis of the chapter biological classification, and we also learned some of the important MCQs in exam point of view under this particular chapter. So in this session, as I told you in the previous session, that from this chapter, it carries a weightage for 5 marks. 2 MCQs will be asked, wherein totally it is of 2 marks, and 1 3-mark question will come from this particular chapter. So here I've put questions which are for 3 marks from this particular chapter, and we will revise them, like how you have to write and all that. So the first question that I've put here is how Ascomycetes are different from Basidiomycetes. So Ascomycetes and Basidiomycetes, they come under the fungi, right? So we got to know that in this particular chapter, that is we learned the 5 kingdom system of classification, wherein we learned Monera, Protista, kingdom fungi, Plantae and Animalia. So fungi, it is made up of 4 classes, that is the Phycomycetes, the Ascomycetes, the Basidiomycetes and Deuteromycetes. Usually the question that is framed when we look at the question papers is write the difference between Ascomycetes and Basidiomycetes, or they frame the question in such a way that how Ascomycetes are different from Basidiomycetes. So whenever a question comes, how is it different from this, or write the difference between this and this, or distinguish between A and B. So when these words come, or illustrate the differences between. So whenever these words come, always make sure that you write it in the form of a column, wherein say for example, this question is how Ascomycetes are different from Basidiomycetes. So you have to find the differences between them. Don't write it in the form of a para. There are chances that you may lose mark and it will not be clear also. So that is why make 2 columns. On one side you write Ascomycetes, and on the other you write Basidiomycetes. And whenever you write the differences also, it should be, say for example, you're writing one point here, the next point should be the opposite of this point that you write. It's not that you write, say for example, I'll say difference between prokaryote and eukaryote. So prokaryote means they do not have a true nucleus, and here you have to write they have cell organelles. Is it the opposite? No. When you write for prokaryotes, they do not have a true nucleus. Then for eukaryotes, you have to write it as they have a true nucleus. Understood? No, it's the opposite that you have to write. Same way here. Ascomycetes, ascospores are produced endogenously. So if you write 3 points, it is enough here. But I've included, that is one more example is also being included here. So ascospores are produced endogenously inside the ascus. That is, it is produced inside a fruiting body which is called as, or a cup-like structure which is called as ascus. So that is one point. Basidiospores are produced exogenously. Can you see the difference here? Exogenously on basidium, this is endogenously inside the ascus. So like this, the differences should be written. Next, Ascomycetes, they produce ascospores and conidia. Basidiomycetes produces what? They produce basidiospores. Ascomycetes reproduce asexually by forming conidia. Basidiomycetes also reproduces asexually only, but what is the difference here? It is either by budding or by formation of spores. Here conidia will not form, either by budding. That's the difference. So examples for Ascomycetes, yeast and penicillium. For Basidiomycetes, it is agaricus and puccinia, whereas yeast is a unicellular fungi, penicillium, then example agaricus is the common mushroom that we consume, and puccinia, it is a rust fungi. So this is about how you have to answer for this question, and this question is quite very important because it keeps repeating now and then. Next, this is also one of the very, very important. Please learn this diagram. It can easily fetch you 3 marks. Usually, they ask this question even for the quarterly test 1 or for midterm, I believe. They had also asked the same thing for midterm, maybe. They had asked the same thing, right? So, if you see the previous question papers, this is repeated quite a number of times. So, please write this diagram, learn this diagram, and always when you draw a diagram or do the labeling, you have to stick on to the NCERT textbook itself. It should not be from some other source. It should be from the NCERT textbook itself. So usually you have to draw. So for 3 marks, 3 labeling is enough, because 4 labeling is what is actually given in your textbook. So 3 if you do, also it is enough. So each labeling will carry half a mark, or 4 labeling if you do, they can consider it as half mark, half for this, half. So half, how many totally? 2 marks for the labeling, that is 1, 2, and 1 mark, or 1 mark is for the way you draw the diagram. And always don't draw the diagram so small. So if the page, you have a A4 sheet, right, which is given for you to write the exam, the booklet, at least half of a page you draw the diagram with clear marking. You mark and you don't put the arrow marks correctly after labeling, then also it will be considered wrong. And whenever you draw a diagram, however neat your diagram is, you don't label, it will be given a 0 mark. So labeling is very, very important here. So you have to draw the head, then comes the collar, then the tail fiber and the sheath. So these are the labelings that are given in the NCERT textbook as well. So accordingly, you have to draw and label. Next question is, name the 3 classes of algae with 1 example for each. Now usually what the students do in a haste is that they just name the 3 classes of algae and they will leave it. They will not write the example, because naming, say for example, 1.5 mark is here. So for half, half, half. How much is this? 1.5. So one example you give, again it is half, half and half. Therefore, how much? 1, 2, 3. That is how the mark is distributed. So you have to give the example as well. So chlorophyceae, the examples are chlorella, chlamydomonas, pyrogyra and cara. Any one you can write. Chlorella, easy to remember. Chlorophyceae, chlorella. One example you can give. Rhodophyceae. So rhodophyceae are the red algae. So you have Porphyra, Gracilaria, Gelidium. Then Phaeophyceae, Dictyota, Laminaria and Sargassum. So these are the examples. Any one example you have to write. So this is also one of the most common question that keeps appearing in the exam. Next question, and this is also one of the most common. So you need to study bacteriophage. So here whatever questions I've put the important ones only. These questions, if you study from this chapter, it's enough, because from that questions only usually the question will appear for the exam. Next, write briefly about the steps involved in sexual cycle of kingdom fungi. There are 3 steps. One is plasmogamy, karyogamy, meiosis. These 3 steps you write. Sometimes they'll just ask, name the steps. Sometimes they'll just say, mention the steps. Then you don't have to explain. Now here, write about the steps, write briefly. So you have to explain. It depends on the way the question is framed. Here, protoplasm. Plasmogamy means protoplasm of 2 motile or non-motile gametes. They fuse together, wherein the protoplasm gets exchanged. That is plasmogamy. Karyogamy means after the fusion of the protoplasm, the nucleus that is there, that should fuse. That is called as karyogamy. Now, after the nucleus gets fused, it will become a zygote, right? That zygote will undergo meiosis to produce haploid spores. That is meiosis. So, plasmogamy, karyogamy and meiosis are the 3 steps. Next question. Give a comparative account of the kingdom Monera and Protista with respect to cell type, cell wall, body organization. Usually they mention the question like this, cell type. So you have to write about the cell type, you have to write about the cell wall, you have to write about the body organization, or sometimes they can include write about mode of nutrition. So that table that is given, if you remember, I think in the next page of your textbook, there is a table, R.H. Whittaker's 5 Kingdom, right? That table, if you study properly, based on that, any question is framed, you can write it easily. So here, Monera, they have asked for Monera and Protista. They can ask for Protista and fungi. or Monera and fungi, like that also they can ask. So here, or in plantain animalia, like that also they can ask. So here, cell type prokaryote. Monera is a prokaryote. Why it is a prokaryote? Because they do not have a true nucleus, wherein the nucleus is not covered by a nuclear membrane. They are a prokaryote. The cell type in Protista is eukaryotic. So they are eukaryotes because their nucleus is covered by a nuclear membrane. Therefore, it is a true nucleus. And also they have membrane bound organelles, whereas Monera, they do not have membrane bound organelle. Next, cell wall. Cell wall, usually most of the Monerans do not have a cell wall. They lack a cell wall, but rarely few of them they have a cell wall, but it is non-cellulosic, meaning it is not made up of cellulose. Instead, it is made up of a sugar, that is polysaccharide, and it is made up of amino acid. Whereas the cell wall in Protista, it is cellulosic. Some may have a cell wall. Protista also, so most of them don't have a cell wall, but some may have a cell wall, and those which have a cell wall, it is made up of cellulose. Okay. So therefore, it is non-cellulosic here, it is cellulosic here. Next, body organization. The cells both are cellular level of organization. It is unicellular. This one is also unicellular itself. So this is what you have to write. So as I told you, this instead of Monera here, they can ask other kingdoms as well. That is, the remaining 3 also can be asked. Next question. This is also one of the very important questions that keep appearing now and then. Cyanobacteria and heterotrophic bacteria. Now, cyanobacteria. Now, what are cyanobacteria? In the previous session, while doing the synopsis, I have told about it. Cyanobacteria are also called as blue-green algae, and they contain chlorophyll pigment in them. Therefore, they are capable of photosynthesizing. So, photosynthesizing means what? They are autotrophs, right? They are autotrophs. So, cyanobacteria and heterotrophic bacteria, though vastly different, have been put together. Put together under which kingdom? Under Monera, right? They are put under Monera. That is, in Monera, again, it is divided into Archaebacteria and Eubacteria. So, these are Eubacteria under Monera. So, they are being clubbed together in Eubacteria of kingdom Monera according to the 5 kingdom system of classification, or according to Whittaker's system of classification. So, is the grouping of the 2 types of taxa in the same kingdom justified? So, the question is, cyanobacteria is an autotroph. It has chlorophyll pigment. Eubacteria, and there are some other bacteria which do not have chlorophyll pigments, but still the cyanobacteria, why have they put the cyanobacteria, and that is the autotrophic bacteria and the heterotrophic bacteria, why have they put them under the kingdom Monera itself? That is, especially in Eubacteria. Apart from Eubacteria, they could have created a third one and put it under that, right? Why have they made only 2, that is Archaebacteria and Eubacteria, and put cyanobacteria and heterotrophic bacteria under Eubacteria? Why have they put them? So, okay, they have put them. So, is it good? Is it okay for you? That is what the question means. So, the answer here is cyanobacteria and heterotrophic bacteria, though vastly different. They are very different. If you look at them, they are very different because cyanobacteria usually they appear like this, like chain-like structures. And the other bacteria, they are normal, rod shape, cocci shape, and all that they will appear to be. See, their mode of nutrition, everything is entirely different. But still, they have been put under the same kingdom for the following reason, because both are unicellular, they are prokaryotic, they do not have a true nucleus, and the nucleus lacks a nucleolus and a nuclear membrane. This is the only reason why they have put it. But you could have asked me now, then they could have put it as Eubacteria only, no? Why they made one more Archaebacteria? Archaebacteria, why? Because those are the bacteria which live in extreme conditions. But these bacteria are not capable of living in extreme conditions. So that was also one difference to make a separate group, that is Archaebacteria and Eubacteria. So moving further, this is also very, very important. That is, draw a label diagram of Nostoc. Here, its diagram, maybe it can carry 2 mark, and this can carry half mark. So this can carry 2 mark. Or it might be 1 mark, 1 mark, 1 mark. It depends on the way the scheme is set, evaluation scheme is set. So you have to be ready for either way. So here only 2 things are there here, easy to remember. One is heterocyst. What is this heterocyst? It helps in fixing atmospheric nitrogen, and mucilaginous sheath. So these two, and draw it properly, you can get marks easily. Bacteriophage and nostoc, one though they will surely ask in the exam. So be prepared for this. Next question. List 6 symptoms of viral diseases in plants. So viral diseases in plants, 6 symptoms. So 6 symptoms means each will carry half mark each of the system. They have totally 3 marks. Mosaic formation, that is patch-like appearance will in the leaf, then rolling of the leaves, curling of the leaves, yellowing of the leaves, the veins of the leaves will get cleared, and the growth of the plant will become stunted. So these 6 symptoms, if you write, 3 marks you will get. And if you're lucky enough, they can ask write 3 symptoms, each symptom carrying 1 mark. That also can be asked. This is also a very, very important question. Can come for 2 marks or 3 marks. 3 marks means they'll ask what is diatomaceous earth. This question will carry 1 mark. And write 2 uses of diatomaceous earth. It will carry 2 mark, 1 mark each. So this is 1 mark. This is 1 mark. So the accumulation of large deposits of silica which were there on the diatoms when the diatoms die, and that accumulation for 100 millions of years will result in the formation of diatomaceous earth. So diatomaceous earth is nothing but silica rich soil because of the dead diatoms deposition. 2 uses are, one is polishing. Why they use it for polishing is it's gritty. Gritty means it's like sand. So sand, it acts like a abrasive. So you can rub it on other surfaces to make the surface clear and smooth. So therefore it is used for polishing. And next it is used as a filtrate because sands also they function like a filtrate. No, same way here. Filtration of oils and syrups. You write so much, you get 3 marks. 3 marks. This is one of the very, very important question and likely to appear in the exam. Next question. Write a note on 3 types of archaebacteria. So 3 types you have to write about: halophiles, thermoacidophiles, methanogens. You have to write a note, or they can ask mention 3 types of archaebacteria. That also can be asked. So halophiles, they live in extreme salt condition. Thermoacidophiles, they live in hot springs. Methanogens, they live in marshy areas and they also live in the gut or intestine of cows, buffaloes, which are the ruminant animals, wherein in the dung of these animals, they produce the methane gas or the biogas. So that is so much you write, you will get marks. And always don't write the biology answers like an art student. No, always make sure it is in points. So points and underline the keywords. Underlining the keywords is very important. Say for example, you write halophytes, underline salty areas, underline hot springs, underline marshy areas, underline gut of ruminant animals. So if the grammatical mistake is there also, looking at the keywords, they'll give you marks. So that is why underlining is very important. Next, list any 4 important characters of kingdom Mycota. They can ask kingdom Mycota. Kingdom Mycota means, don't get confused, it is nothing but fungi. Kingdom fungi. One is cell wall is made up of chitin and polysaccharides. Body is made up of, they have long thread-like structures, and those thread-like structures are called what? They're called hyphae. That is, long thread-like structures will be there, very thin, slender thread-like structures they'll have. And the network of these thread-like structures, which will result in the formation of a mesh-like appearance, it is called as mycelium. So you have to write about hyphae and mycelium. Forget to write how you need to create a sentence. What I told you, underline the keywords. Underline hyphae, underline mycelium there. So picking the keywords, marks will be given. And they have cenocytic hyphae. That is, some with septa, and they are, or that is some without septa and some with septa. What is cenocytic hyphae? Cenocytic hyphae means septas will not be present, therefore they'll exhibit a multinucleate condition in the hyphae. That is called cenocytic hyphae. Septa means they'll have cross walls in their hyphae like this, wherein in each of the septa they'll have a single nucleus distributed like this. So that's the difference. And they are saprophytic. Saprophytic means fungi usually grow on dead and decaying organic matter. Any food item or any vegetable or bread, for example, you keep it in the fridge and you forget it. After a few days, when the food rots, you can find fungi growing on it. For example, rhizospores and all that. So they are saprophytic in nature. So any 4 points you write, you will get marks here. Or if it is for 2 marks, they'll ask any 4 points or any 3 points you can write depending on how the question is framed. So these are some of the very, very important questions that I've put here, which are likely to appear in the exam, that is for your final exams. So concentrate on these and study well. So see you in the next revision session, wherein we'll take up a new chapter and discuss some of the important questions under that particular chapter. So see you in the next session. Thank you.